Hello, welcome to a quick presentation titled Rock Solid, a life force for the Earth, where we will tackle topics about the ecosystem, particularly in land. Let's start things off with the biogeochemical cycle, the sedimentary cycle. It is the weathering of existing rocks, with Earth's crust as the reservoir, then the erosion of minerals, transport, deposition, and burial. Materials that are moved and deposited into a new location are called sediments. There are three basic examples of sediments. They are rocks, minerals, and the remains of plants and animals. The most common sedimentary rocks are sandstones, limestones, and shales. The more cycle sediments undergo, the more mature the material will become. An example of sedimentary cycle is phosphorus cycle. The phosphorus cycle is a sedimentary cycle that shows the movement of phosphorus through the lithosphere, hydrosphere, and biosphere. Most of Earth's phosphorus is in sedimentary deposits released through numerous sedimentary cycles. But what is phosphorus? Phosphorus is a mineral needed for plant and animal growth and microorganisms. This is a beneficial and essential mineral as it functions to repair and produce genetic building blocks such as DNA and RNA. Here is a simple illustration of the phosphorus cycle. Each process undergoes simultaneously with one another, creating a continuous cycle that moves phosphorus from place to place. From weathering and erosion from mountains and rocks to decomposition of plants and animals that goes through the soil, which then transfers through bodies of water, which will then undergo a phosphate rock formation in order to create new rocks for the phosphorus cycle to continue. So there you have it for the phosphorus cycle.
The term anthropogenic, as defined by the dictionary, is something relating to or resulting from the influence of human beings on nature. Therefore, anthropogenic effects refers to the changes brought about by human activities. These changes, more often than not, are negative in nature. One such change is called anthropogenic eutrophication. Anthropogenic because this eutrophication is encouraged by human activities. Eutrophication is characterized by the algal growth in bodies of water, more commonly fresh water. The algal growth is due to the overwhelming amount of nutrients in the water. One such nutrient involved is phosphorus. Eutrophication has many causes. However, the most notable ones are fertilizers that is carried off by runoff water from farmlands and the improper disposal of untreated sewage water. The most noticeable damage caused by eutrophication is the greenish color that it gives to the water and the accompanying odor. But more importantly, it can also affect human and animal life. Most aquatic life cannot live in affected waters, while the odor and color renders the nearby area unsuitable for tourism. And on the worst case scenario, it may even pose a threat to the health of nearby communities. There is no instant solution for eutrophication. However, we can employ prevention measures in order to protect unaffected areas and help affected areas recover.
So, what are invasive species? Invasive species are organisms such as plants, animals, fungi, etc., that are not native to their environment, with the goal of taking over the current environment through all means necessary. Invasive species compete with native species for food supply and territory, cause extensive damage to the health of plants, animals, and even humans, and are typically caused by human activity, often unintentionally. In the next few slides, I will show examples of invasive species that have caused issues around the world. European green crabs are brought to many ecosystems by being carried by ships in ballast water and in some cases, being sold as fish bait. European green crabs as an invasive species has established populations of both coasts on the North American continent, in South America, Australia, South Africa, and Japan. In the Philippines, water hyacinths have taken over the surface of rivers, most notably the Pasig River and Laguna de Bay, which prevented aquatic organisms from obtaining oxygen and competed with the native flora and algae that thrive there. It also poses a problem for people who use boats in the river since the water hyacinth stems and leaves get in the way of the boat and make it difficult for them to pass through. So, as regular people, how do we prevent invasive species from spreading? The best way to fight invasive species is to prevent them from occurring in the first place. Firstly, don't release your pets into the wild, but if you do that, it might, introduce another set of invasive species. Secondly, ensure that the plants you're growing are not invasive. And lastly, express your awareness about the harmful effects of invasive species.
Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads the DNA sequence. DNA methylation. DNA methylation works by adding a chemical group to DNA. Typically, this group is added to specific places on the DNA, where it blocks the proteins to attach to the DNA to read the gene. This chemical group can be removed through a process called demethylation. Typically, methylation turns genes off and demethylation turns genes on. Histone modification. DNA wraps around protein called histones. DNA wrapped tightly around histones cannot be accessed by protein that read the gene. Some genes are wrapped around histones and are turned off, while some genes are not wrapped around histones and turned on. Chemical group can be added or removed from histones and change whether a gene is unwrapped or wrapped on or off. Non-coding RNA. Your DNA is used as instructions for making coding and non-coding RNA. Coding RNA is used to make proteins, and non-coding RNA helps control gene expressions by attaching coding RNA along with certain proteins to break down the coding RNA so that it cannot be used to make proteins. Non-coding RNA may also recruit proteins to modify histones to turn off and on. Epigenetic changes begin before you are born. All cells have the same genes but look and act differently. As you grow and develop, epigenetics help determine which function a cell will have. For example, whether it will become a heart cell, nerve cell, or skin cell. For example, nerve cell versus muscle cell. Your muscle cells and nerve cells have the same DNA but work differently. Nerve cells can transport information to other cells in your body. A muscle cell has a structure that aids your body's ability to move. Epigenetics allow muscle cell to turn on genes to make proteins important for its job and turn off genes important to nerve cells' job. Epigenetic changes throughout your life. Your epigenetic at birth is not the same as epigenetic during childhood or adulthood. A study of newborn versus 26-year-old versus 103-year-old. DNA methylation at millions of sites were measured in a newborn, 26-year-old, and 103-year-old. The level of DNA methylation decreases with age. A newborn had the highest DNA methylation. No, the 103-year-old had the lowest DNA methylation, and the 26-year-old had DNA methylation level between the newborn and the 103-year-old. Not all epigenetic change are permanent. Some epigenetic changes can be added or removed in response to changes in behavior or environment. Smoking can result in epigenetic changes. For example, at certain parts of the AHRR gene, smokers tend to have less DNA methylation than non-smokers. The difference is greater for heavy smokers and long-term smokers. After quitting smoking, former smokers can begin to have increased DNA methylation genes at these genes. Eventually, they can reach levels similar to those of non-smokers. In some cases, this can happen in under a year. Germs can change your epigenetics to weaken your immune system. This helps the germ survive. For example, Mycobacterium tuberculosis. Mycobacterium tuberculosis causes tuberculosis. Infection with these germs can cause changes to histone in some of your immune cells that results in turning off the IL-1B gene. Turning off the gene weakens your immune systems and improves the survival of Mycobacterium tuberculosis.